Hello there and welcome to Deep Sleep Seas. I'm so glad you found your way here tonight. Perhaps you've had a long day, or maybe you're the kind of person who finds comfort in the deep mysteries of the ocean rather than counting waves rolling onto shore. Either way, tonight's journey is for you. You have nothing to do but rest. Let your mind settle, your breathing slow and your body grow heavy as if the weight of 1,000 fathoms has gently pressed down upon you. Because tonight, we're not simply exploring the surface of the sea, we're descending into one of the ocean's strangest and most profound mysteries, a place where the water itself seems to hold its breath. Deep beneath the sunlit waves, in the middle layers of the tropical and temperate oceans, there exist vast zones where oxygen nearly vanishes from the water. These are the oxygen minimum zones, strange twilight regions where the very substance that most life depends upon becomes so scarce that survival seems impossible. Yet life persists here, transformed and adapted in ways that challenge everything we think we know about the requirements for existence. These are not lifeless voids. They are inhabited realms governed by their own rules, shaped by invisible forces, and home to creatures that have learned to breathe what others would call suffocation. The ocean you see from shore, rolling with whitecaps and breathing salt spray into the wind, holds within it layers upon layers of hidden worlds. The surface sparkles with oxygen drawn from the air above. Fish leap, dolphins arc through waves, and seabirds dive for silver schools of herring and mackerel. But descend a few hundred meters, past the reach of sunlight, into the dim blue twilight of the mesopelagic zone, and you enter a different ocean entirely. Here, in certain parts of the world, oxygen levels drop to concentrations so low that they would be lethal to most animals. Some regions hold oxygen levels measured not in parts per million, but in parts per billion, concentrations so vanishingly small that they hover at the edge of detection, flirting with complete anoxia. These oxygen minimum zones are not random accidents of chemistry. They form through a delicate interplay of physical and biological processes, layered one atop another like sediment accumulating on the seafloor. Warm surface water heated by tropical sunlight creates a barrier, a thermocline that prevents mixing with the cold, oxygen-rich waters below. Meanwhile, from the sunlit surface, a constant rain of organic matter drifts downward, fragments of dead plankton, fecal pellets, discarded mucus houses from drifting lavaceans, marine snow in all its forms, settling through the water column like ash from an invisible fire. As this material descends, bacteria and other decomposers feast upon it, consuming oxygen in the process of breaking down the organic carbon. Respiration depletes what little oxygen exists at these middle depths, and with poor circulation to replenish it, the oxygen concentration plummets. The result is a layer sandwiched between the well-oxygenated surface and the cold, oxygen-rich deep. It's a zone of scarcity, a band of near suffocation that stretches horizontally for thousands of miles in some parts of the ocean. The largest of these zones exist in the eastern tropical Pacific, off the coasts of South America and Central America, where upwelling brings nutrients to the surface and fuels enormous blooms of phytoplankton. When those blooms die and sink, they feed the microbial communities below, creating one of the most oxygen-depleted regions on Earth. Another major zone lies in the Arabian Sea, where seasonal monsoons drive intense productivity and create similar conditions. Smaller zones exist off the coast of West Africa and in other tropical regions where the right combination of warmth, productivity, and poor ventilation conspire to starve the water of breath. To an animal that depends on oxygen, these zones are barriers as real as any physical wall. Tuna and swordfish, built for speed and endurance, cannot linger here. Their metabolisms demand too much. They must pass through quickly or avoid these zones altogether. Many fish navigate around oxygen minimum zones, staying in the oxygenated surface waters or diving deeper to the well-mixed abyssal plains below. Some creatures, like Humboldt squid, migrate vertically through these zones each day, ascending to feed in richer waters at night and descending into the oxygen minimum zone during the day to avoid predators. They endure the hypoxia as a trade-off, a temporary discomfort worth the safety it provides. But there are others, stranger and more specialized, that have made these oxygen-starved waters their permanent home. They are the residents of the suffocating sea, creatures that have evolved not to avoid low oxygen, but to embrace it, to thrive where others cannot survive. And in doing so, they've discovered something remarkable. In a world without breath, there are fewer mouths to compete with, fewer predators to fear, and an abundance of food drifting down from above that most animals cannot stay long enough to claim. The oxygen minimum zone, for all its harshness, is also a refuge. Before we descend any further, if you find these stories soothing, consider subscribing so you don't miss the next journey into the deep. And if you're still awake enough, let me know where in the world you're listening from and what time it is where you are. Now let us sink deeper, past the edge of light, into the layered shadows where oxygen fades and a different kind of ocean begins. Oxygen minimum zones do not appear suddenly. They form gradually, built layer by layer through processes that span days, seasons, and centuries. To understand how they come to exist, we must first understand what oxygen means in the ocean and how it moves through the water. At the surface, the ocean breathes. Waves break and churn, folding air into water, and oxygen from the atmosphere dissolves into the sea. Cold water holds more dissolved oxygen than warm water, which is why polar seas are rich in oxygen while tropical waters hold less. The colder the water, the more readily it absorbs oxygen molecules from the air, storing them in solution, where marine life can extract them through gills, through skin, through specialized membranes designed to pull breath from liquid. Photosynthesis adds to this oxygen supply. In the sunlit surface layer, the euphotic zone, phytoplankton photosynthesize during daylight hours, splitting water molecules and releasing oxygen as a byproduct. This is the breath of the ocean's green pastures, the invisible exhalation of microscopic drifters that collectively produce more oxygen than all the forests on land. During the day, oxygen levels near the surface can even become supersaturated, holding more oxygen than equilibrium with the atmosphere would suggest, a gift from the plankton that drift in their billions through every liter of seawater. But this oxygen does not spread evenly through the depths. The ocean is stratified, layered like a cake, with warm, light water floating atop colder, denser water below. Between these layers lies the thermocline, a zone of rapid temperature change that acts as a barrier to vertical mixing. Warm surface water does not easily mix with cold, deep water. The density difference creates a stable layering, and without mixing, oxygen cannot be replenished at depth through contact with the atmosphere or through the sinking of oxygenated surface water. Now imagine organic matter beginning its long descent. A microscopic diatom dies in the surface waters, its silica shell and organic contents sinking slowly through the water column. A copod defecates, releasing a fecal pellet that tumbles downward. A jellyfish expires, its gelatinous body drifting like a ghost through the thermocline. A larvacean, a tiny filter-feeding creature, abandons its mucus house after clogging it with trapped particles, and the house, laden with concentrated food, begins to sink. This is marine snow, a constant gentle blizzard of organic particles falling from the productive surface layers toward the deep. The particles that make up marine snow are diverse in size, composition, and sinking rate. Some are individual cells or fragments of cells, small and slow sinking, taking weeks or months to descend through the water column. Others are aggregates, clusters of particles stuck together by mucus and other sticky substances, larger and faster sinking, completing their journey to the deep in days or weeks. The largest particles, such as dead jellyfish or salt chains, can sink at rates of hundreds of meters per day, reaching the deep ocean quickly and delivering pulses of organic matter to the seafloor. As marine snow descends through the water column, it changes. Bacteria colonize the particles, breaking down the organic matter and consuming oxygen in the process. Zooplankton feed on the particles, fragmenting them and repackaging them into fecal pellets. The particles shrink, losing mass and nutrients as they are consumed and remineralized. By the time marine snow reaches the oxygen minimum zone, much of its most labile organic matter has already been consumed, leaving behind the more refractory compounds that are harder for microbes to break down. But even this refractory material is valuable in the oxygen minimum zone, where food is scarce and competition is reduced. The vampire squid, with its slow metabolism and patient lifestyle, can extract nutrition from marine snow that other animals would find unpalatable. The bacteria and archaea that inhabit the oxygen minimum zone have specialized enzymes that can break down complex organic compounds, extracting energy from molecules that would be inaccessible to most organisms. In this way, marine snow sustains life in the oxygen minimum zone, providing a continuous supply of carbon and energy that fuels the microbial communities and supports the food web. The flux of marine snow to the oxygen minimum zone varies with season, with productivity in the surface ocean, and with changes in ocean circulation. During periods of high productivity, when phytoplankton blooms are intense, the flux of organic matter increases, delivering more food to the oxygen minimum zone and intensifying oxygen consumption. During periods of low productivity, the flux decreases, and oxygen levels may recover somewhat as respiration slows. This variability creates a dynamic environment where oxygen levels fluctuate in response to the pulses of organic matter that rain down from above. As this material descends, it passes through communities of bacteria and archaea that cling to the particles or drift freely in the water, waiting. These microbes are decomposers, breaking down the organic carbon through respiration, using oxygen to convert the energy stored in dead tissue back into forms they can use. Each act of respiration consumes oxygen and releases carbon dioxide. Multiply this by trillions of microbial cells across thousands of cubic kilometers of water, and the effect becomes profound. Oxygen levels begin to drop. In most parts of the ocean, this oxygen depletion is countered by circulation. Deep water formed at high latitudes, where cold, oxygen-rich water sinks and spreads across the ocean floor, eventually upwells or mixes back toward the surface, replenishing the oxygen that respiration has consumed. But in the middle depths of certain tropical regions, circulation is sluggish. The water is trapped, isolated from both the oxygenated surface above and the well-ventilated deep below. It sits, stagnant or near stagnant, while organic matter continues to rain down and microbes continue to consume oxygen. Over time, oxygen concentrations fall to levels that would be considered hypoxic, then anoxic. The depth at which this occurs varies by location, but typically oxygen minimum zones form between 200 and 1500 meters below the surface. This is the mesopelagic zone, the twilight zone, where sunlight has faded to a dim blue glow or vanished entirely, but where the water is still warmer than the abyssal cold below. The oxygen concentration may drop below 2 milligrams per liter, below 1 milligram per liter, in some places below detection limits, effectively reaching zero. These are the core regions of oxygen minimum zones, the heart of the suffocation. The boundaries of these zones are not sharp lines but gradients. At the upper edge, oxygen levels decline gradually as you descend from the well-mixed surface layer. This transition zone, the oxycline, is where oxygen concentration drops steeply over a short vertical distance. Pass through the oxycline, and you enter the oxygen minimum zone proper. At the lower edge, oxygen levels begin to rise again as you descend into the colder, better ventilated deep ocean. The oxygen minimum zone is thus a lens, a layer of low oxygen water suspended between two better oxygenated regions, held in place by the architecture of temperature, density, and circulation. What determines where these zones form? Geography plays a role. Oxygen minimum zones are most pronounced along the western coasts of continents in the tropics, where upwelling brings nutrient-rich water to the surface and drives intense phytoplankton blooms. The eastern tropical Pacific, off the coasts of Peru, Ecuador, and Mexico, hosts one of the largest and most intense oxygen minimum zones on Earth. Here, the combination of high productivity, warm stratified water, and limited ventilation creates conditions where oxygen can be completely exhausted from the water column. The Arabian Sea experiences similar conditions, driven by monsoon-driven upwelling and high surface productivity. Other factors influence oxygen minimum zone formation as well. The shape of ocean basins, the patterns of wind-driven and thermohaline circulation, the depth of the thermocline, the rate of organic matter export from the surface. All of these variables interact to determine whether an oxygen minimum zone will form and how intense it will become. In some regions, oxygen minimum zones are permanent features, persisting year-round for millennia. In others, they are seasonal, forming during periods of intense upwelling and productivity, then relaxing when circulation improves or productivity declines. The water in an oxygen minimum zone is not still. It flows slowly, following the large-scale circulation patterns of the ocean. Water masses move through these zones, carrying with them the chemical signatures of their history, the oxygen they started with, the organic matter they've encountered, the microbial transformations they've undergone. Some water enters an oxygen minimum zone already low in oxygen, having traveled far from its source in the polar regions and aged along the way. its oxygen gradually consumed by respiration. Other water is ventilated more recently, but is overwhelmed by the intense oxygen demand created by high productivity above. The result is a layered structure within the oxygen minimum zone itself. The core, where oxygen is lowest, may be only a few hundred meters thick, but it is surrounded by gradients of increasing oxygen concentration above and below. These gradients are not uniform. They can vary horizontally as well as vertically, creating patchiness in oxygen concentration across space. A submersible descending through an oxygen minimum zone might encounter pockets of slightly higher oxygen, intrusions of better oxygenated water mixing in from nearby regions, or eddies that bring transient relief before the suffocation closes in again. For the organisms that live here, these gradients are everything. The difference between 5 micromolar oxygen and 0 micromolar oxygen can be the difference between survival and death, between a niche that can be exploited and a void that cannot be inhabited. Life in oxygen minimum zones is life on a knife's edge, constantly balancing the need for energy against the scarcity of the resource that makes energy extraction possible. And yet, as we will see, life has found ways to thrive even here, in the thinnest margins of habitability, where breath is measured in molecules and survival is an act of biochemical ingenuity. The ocean continues its slow circulation, indifferent to the struggles of the creatures within it. Water flows, particles sink, bacteria respire, and oxygen fades into scarcity. The oxygen minimum zone persists, a stable feature of the ocean's architecture, a layer of near suffocation, maintained by forces that have operated for thousands of years and will continue for thousands more. In the heart of an oxygen minimum zone, where oxygen concentrations hover near zero, most animals cannot survive. The metabolic machinery of fish, squid, crustaceans, and other active creatures requires oxygen to function. Without it, they cannot generate the energy needed to swim, to hunt, to flee, to maintain the cellular processes that keep them alive. And yet, the oxygen minimum zone is not empty. It is inhabited, populated by creatures that have learned to make do with almost nothing, or by those that have found alternative chemistries to fuel their existence. Consider the vampire squid, one of the most remarkable residents of these oxygen-starved waters. Its name, vampirotooth, is infernalis, translates to vampire squid from hell, a dramatic title for a creature that is in truth quite gentle. It does not suck blood. It does not hunt living prey. Instead, it drifts through the oxygen minimum zone, feeding on marine snow, collecting detritus with specialized filaments that it extends like fishing lines into the darkness. The vampire squid has adapted to life in the oxygen minimum zone so completely that it can survive at oxygen saturations as low as 3%, levels that would kill most cephalopods within minutes. How does it accomplish this? Through a suite of remarkable adaptations that together make it one of the most oxygen-efficient animals in the sea. Its metabolic rate is the lowest of any cephalopod, a deliberate slowing of life's processes to conserve the meager oxygen available. Its blue blood contains hemocyanin, a copper-based oxygen-carrying protein that binds oxygen more efficiently than the hemocyanin of other cephalopods, wringing every molecule from the depleted water. Its gills are large, with an immense surface area relative to its body size, maximizing the extraction of oxygen from each liter of water that passes over them. The blood-to-water diffusion distance across the gill membranes is thin, allowing oxygen to pass rapidly from water to blood. The vampire squid's body is gelatinous, weak-muscled, almost insubstantial. It lacks the powerful musculature that other squids use for jet propulsion, the rapid escape responses, the aggressive hunting. Instead, it relies on neutral buoyancy, its tissues carefully balanced with the density of seawater through the use of ammonium-rich compounds that lighten its body without requiring energy expenditure. It can swim, using its fins to glide gracefully through the water, but it does not waste energy on speed or struggle. It moves with the slow deliberation of a creature that has learned to ration every breath. Its eyes are enormous relative to its body, the largest eyes in proportion to body size of any animal in the ocean. These eyes are not for hunting in the traditional sense, but for detecting bioluminescence, the faint flickers of light produced by other organisms in the darkness. The vampire squid watches, alert to the presence of marine snow aggregates, to potential mates, to the rare predator that might venture into its low-oxygen refuge. When threatened, it does not flee with the rapid jets of a typical squid. Instead, it performs a defensive posture called the pineapple posture, inverting its webbed arms over its body to expose spiny cirri, making itself difficult to grasp or swallow. It can also produce bioluminescent mucus, glowing clouds that confuse and distract predators, giving the vampire squid time to drift away into the darkness. The vampire squid is not alone in the oxygen minimum zone, though it is perhaps the most iconic resident. Other creatures have found their own ways to survive here. The giant red mysid, a shrimp-like crustacean, has highly developed gills and an efficient circulatory system that allows it to extract up to 90% of the oxygen from the water it inhales. Certain species of jellyfish, with their low-energy gelatinous bodies and minimal metabolic demands, drift through oxygen minimum zones, feeding on detritus and small prey. These creatures require so little energy that they can survive on the faintest whisper of oxygen, their lives slowed to a pace that matches the stillness of the water around them. Some fish make brief forays into the oxygen minimum zone, not as residents but as commuters. Humboldt squid, also known as jumbo squid, are powerful predators that hunt in the oxygen-rich surface waters at night, but descend into the oxygen minimum zone during the day. They endure the hypoxia, tolerating oxygen levels that would incapacitate other fish because the oxygen minimum zone offers them protection from larger predators. Tuna, swordfish, and marlin cannot follow them into these depths, and so the Humboldt squid gains a refuge, a place to rest during daylight hours before ascending again under cover of darkness to feed. Other animals migrate vertically through the oxygen minimum zone each day, part of the largest mass migration on the planet. Zooplankton, small fish, and squid rise from the deep during the night to feed in the rich surface waters, then descend at dawn to avoid predators. For many of these migrants, the journey takes them through the oxygen minimum zone, a passage they must endure twice daily. They have evolved to tolerate brief exposure to hypoxia, holding their breath in a sense, or slowing their metabolism temporarily to survive the crossing. The oxygen minimum zone becomes a gauntlet, a trial that must be passed to access the rewards of the surface. For some creatures, the oxygen minimum zone is less a barrier and more an opportunity. With fewer predators able to tolerate the low oxygen, the zone offers a relatively safe environment for those that can adapt. The detritus that rains down from above accumulates here, providing a steady food source that is less contested than in the oxygen-rich waters above or below. The vampire squid, with its detritivorous lifestyle, exploits this niche, feeding on a resource that other cephalopods, built for active hunting, cannot efficiently access. In the economy of the oxygen minimum zone, slow and patient wins the race. The adaptations required to live in such an environment are not trivial. They represent millions of years of evolutionary refinement, trade-offs between the benefits of safety and food availability, and the costs of living in perpetual near suffocation. The vampire squid, for instance, sacrifices speed and strength for oxygen efficiency. It cannot hunt active prey, cannot escape rapidly from fast-moving predators, and likely has a slower growth rate and longer generation time than its relatives in oxygen-rich waters. But in the oxygen minimum zone, these trade-offs are worth it. Here, the vampire squid finds a world where its adaptations give it an edge, a place where being slow and patient is not a disadvantage, but a strategy for survival. The existence of these creatures in the oxygen minimum zone challenges our understanding of the limits of life. We tend to think of oxygen as a fundamental requirement, as essential as water or carbon, and in a sense, it is. But the oxygen minimum zone shows us that essential is a relative term, that life can persist at concentrations of oxygen so low they are barely measurable, that the boundary between sufficient and insufficient is not a line but a gradient, and that evolution is endlessly creative in finding ways to push that boundary further and further toward the edge of impossibility. The vampire squid, drifting through the darkness with its enormous eyes and gossamer body, is a testament to life's resilience, a creature that has learned to breathe the thinnest air and still call it home. While the vampire squid and other macroscopic animals draw our attention, the true masters of the oxygen minimum zone are invisible to the naked eye. These are the microbes, bacteria and archaea that thrive in the low oxygen and anoxic waters, performing biochemical transformations that are essential to the functioning of the global ocean and the planet itself. In the oxygen minimum zone, microbes outnumber all other forms of life by orders of magnitude, and their metabolic activities shape the chemistry of the water, influencing everything from nutrient availability to greenhouse gas production. In the presence of oxygen, Life is relatively straightforward. Organisms use oxygen as an electron acceptor in cellular respiration, breaking down organic matter and extracting energy in the process. This is aerobic respiration, the metabolic pathway that powers most of the visible life on Earth. But in the oxygen minimum zone, where oxygen is scarce or absent, microbes must turn to alternative strategies. They become anaerobic, using other chemicals as electron acceptors, and in doing so, they drive some of the most important biogeochemical cycles in the ocean. One of the most significant processes occurring in oxygen minimum zones is denitrification, the stepwise reduction of nitrate to nitrogen gas. Nitrate, abundant in the oxygen minimum zone, serves as an electron acceptor for bacteria when oxygen is unavailable. The bacteria take nitrate molecules and reduce them, first to nitrite, then to nitric oxide, then to nitric oxide, then to nitric oxide, and finally to nitrogen gas. Each step releases energy that the bacteria can use to power their cellular processes. The end product, nitrogen gas, escapes from the water as bubbles or dissolved in the seawater, eventually making its way back to the atmosphere. Denitrification is not a trivial process. It represents a loss of bioavailable nitrogen from the ocean, nitrogen that could otherwise be used by phytoplankton to build proteins and nucleic acids. The ocean's nitrogen budget is a delicate balance between nitrogen fixation, which converts atmospheric nitrogen gas into forms that life can use, and nitrogen loss through denitrification and related processes. Oxygen minimum zones, though they occupy only a small fraction of the ocean's volume, account for a disproportionate amount of global nitrogen loss. Some estimates suggest that 30 to 50% of oceanic nitrogen loss occurs in these zones, making them critical regulators of ocean fertility and productivity. The microbial communities responsible for denitrification are diverse. Some bacteria carry out the entire denitrification pathway, reducing nitrate all the way to nitrogen gas. Others perform only one or two steps, reducing nitrate to nitrite or nitrite to nitrogen gas, but not the complete transformation. These partial denitrifiers rely on other members of the microbial community to complete the process, creating a network of metabolic dependencies that spans the entire oxygen minimum zone. The composition of denitrifying communities changes with oxygen concentration, with depth, and over time, as different species or strains become dominant depending on the availability of substrates and the environmental conditions. Another important process in oxygen minimum zones is anammox, short for anaerobic ammonium oxidation. This process is performed by specialized bacteria that combine ammonium and nitrite to produce nitrogen gas and water without any need for oxygen. Anammox bacteria are slow-growing and difficult to culture, and they were only discovered relatively recently, despite their importance to the nitrogen cycle. In the oxygen minimum zone, anammox can be a major pathway for nitrogen loss, sometimes exceeding the contribution of traditional denitrification. Anammox bacteria require both ammonium and nitrite to function, which means they depend on other microbial processes to supply these substrates. Ammonium can come from the breakdown of organic matter, from the excretion of animals, or from a process called dissimilatory nitrate reduction to ammonium, or DNRA, in which bacteria reduce nitrate to ammonium instead of to nitrogen gas. Nitrite can come from the incomplete reduction of nitrate by denitrifiers or from the oxidation of ammonium by nitrifying bacteria. The interplay between these processes creates a complex web of nitrogen transformations, each one influencing the others, each one shaped by the availability of oxygen, organic matter, and other environmental factors. In some parts of the oxygen minimum zone, particularly where sulfide is present, sulfur-oxidizing bacteria play a critical role. These bacteria oxidize hydrogen sulfide, using nitrate or nitrite as electron acceptors, and in doing so, they produce sulfate and contribute to the nitrogen cycle. One group of sulfur-oxidizing bacteria, known as SUP05, is particularly abundant in oxygen minimum zones, and plays a key role in coupling sulfur and nitrogen cycling. These bacteria can respond rapidly to changes in sulfide availability, blooming when sulfidic conditions develop and declining when sulfide disappears. The presence of sulfide in an oxygen minimum zone is a sign of extreme anoxia. Under most low oxygen conditions, sulfate-reducing bacteria do not dominate, and sulfide does not accumulate. But in the most oxygen-depleted cores of oxygen minimum zones, or in enclosed basins where ventilation is particularly poor, sulfide can appear, creating what are sometimes called sulfidic oxygen minimum zones. These are rare, but when they occur, they represent an even more extreme environment, one where the chemistries of ancient oxygen-free oceans momentarily reassert themselves in the modern sea. Microbes in oxygen minimum zones do not work in isolation. They form communities, structured by the availability of resources, by competition and cooperation, by the physical environment. Some microbes attach to sinking particles, riding them down through the water column and feeding on the organic matter they carry. These particle-associated communities are hotspots of microbial activity, with higher concentrations of bacteria and higher rates of respiration and nutrient cycling than the surrounding free-living communities. Other microbes drift freely in the water, scavenging dissolved organic matter or relying on chemolithotrophy, the use of inorganic chemicals as energy sources to build biomass from carbon dioxide. The activity of these microbial communities has consequences that ripple far beyond the oxygen minimum zone. The nitrogen gas produced by denitrification and anammox escapes to the atmosphere, altering the global nitrogen budget and influencing the availability of nitrogen for photosynthesis in the surface ocean. The nitrous oxide produced as an intermediate in denitrification is a potent greenhouse gas with a warming potential hundreds of times greater than carbon dioxide. Oxygen minimum zones are significant sources of nitrous oxide to the atmosphere, contributing to climate change and linking the health of the ocean to the temperature of the planet. The microbes of the oxygen minimum zone are also part of the carbon cycle. As they break down organic matter, they release carbon dioxide, which dissolves in the seawater and eventually makes its way back to the atmosphere or is transported to the deep ocean by circulation. The efficiency with which organic carbon is remineralized in the oxygen minimum zone influences how much carbon is sequestered in the deep sea and how much returns to the surface to be recycled by phytoplankton. In this way, the invisible activity of bacteria in the oxygen minimum zone influences the ocean's role as a carbon sink, a buffer against the rising levels of carbon dioxide in the atmosphere. For all their importance, the microbes of the oxygen minimum zone remain poorly understood. Sampling them is challenging. Traditional methods of collecting water samples can introduce oxygen contamination, making it difficult to study anaerobic processes in their natural state. New technologies, including in situ sensors and autonomous sampling devices, are beginning to overcome these challenges, but much remains to be learned about the diversity, activity, and ecological roles of oxygen minimum zone microbes. Each new study reveals unexpected complexity, new metabolic pathways, new species, new interactions. The microbial world of the oxygen minimum zone is vast, ancient, and still largely unexplored. Nitrogen is a paradox in the ocean. It is one of the most abundant elements in the atmosphere, making up 78% of the air we breathe, yet it is one of the most limiting nutrients for ocean life. This is because nitrogen gas, NiS, is chemically inert, a molecule so tightly bonded that most organisms cannot break it apart and use it. To enter the biological cycle, nitrogen gas must first be fixed, converted into reactive forms like ammonium or nitrate that phytoplankton can incorporate into their cells. This fixation is carried out by specialized bacteria and archaea, and it is a slow, energy-intensive process that limits how much nitrogen is available for life in the sea. Once nitrogen is fixed, it enters a complex cycle of transformations, passing through various chemical forms as it is taken up by phytoplankton, passed through the food web, excreted, decomposed, and recycled. Some of this nitrogen eventually makes its way to the oxygen minimum zone, carried there by sinking organic matter or transported by ocean currents. And it is here, in the low oxygen and anoxic waters, that much of the ocean's nitrogen is lost, converted back into nitrogen gas, and returned to the atmosphere, completing the cycle. The loss of nitrogen in oxygen minimum zones has profound implications for ocean productivity. Nitrogen is often the nutrient that limits phytoplankton growth in much of the ocean. When nitrogen is lost through denitrification and anammox in oxygen minimum zones, it reduces the overall pool of bioavailable nitrogen in the sea, which in turn limits how much phytoplankton can grow, how much carbon dioxide can be absorbed from the atmosphere, and how much food is available for the marine food web. The nitrogen cycle is thus a critical control on the ocean's biological productivity and its role in regulating the Earth's climate. The balance between nitrogen fixation and nitrogen loss is not static. It shifts over time, influenced by changes in ocean circulation, temperature, productivity, and oxygen levels. During periods when oxygen minimum zones expand, nitrogen loss increases, which can reduce ocean productivity and alter the chemistry of the sea. During periods when oxygen minimum zones contract, nitrogen loss decreases, allowing bioavailable nitrogen to accumulate and potentially enhancing productivity. This balance is sensitive to external forcing, to the warming of the climate, the addition of nutrients from land, the patterns of wind and current that drive upwelling and mixing. One of the most important aspects of nitrogen loss in oxygen minimum zones is the production of nitrous oxide, N-thiol, a greenhouse gas that is produced as an intermediate step in denitrification. Not all of the nitrate that enters the denitrification pathway is fully reduced to nitrogen gas. Some is converted only to nitrous oxide, which can then escape from the water and enter the atmosphere, where it contributes to global warming and also plays a role in the destruction of stratospheric ozone. Oxygen minimum zones are among the largest natural sources of nitrous oxide to the atmosphere, and the amount of nitrous oxide they produce is sensitive to the intensity and extent of low oxygen conditions. In recent decades, as climate change has begun to warm the ocean and expand oxygen minimum zones, there is concern that nitrous oxide production may increase, creating a positive feedback loop. Warming leads to deoxygenation, which expands oxygen minimum zones, which increases nitrogen loss and nitrous oxide production, which contributes to further warming. This feedback is not yet fully understood, and its magnitude is uncertain, but it represents a potential tipping point in the Earth system, a way in which changes in the ocean could amplify changes in the climate. The nitrogen cycle in oxygen minimum zones is also linked to the carbon cycle. When nitrogen is lost from the ocean, it reduces the capacity of phytoplankton to fix carbon dioxide through photosynthesis. Less nitrogen means less phytoplankton growth, which means less carbon dioxide is absorbed from the atmosphere and less organic carbon is exported to the deep sea. In this way, the nitrogen cycle in oxygen minimum zones influences the ocean's ability to act as a carbon sink, to buffer the rising levels of carbon dioxide in the atmosphere. But the relationship is not simple. The microbes that perform denitrification and amamics are themselves consuming organic carbon as they break it down, releasing carbon dioxide in the process. The balance between carbon fixation in the surface ocean and carbon remineralization in the oxygen minimum zone determines how much carbon is ultimately sequestered in the deep sea. If oxygen minimum zones expand and nitrogen loss increases, the net effect on the ocean's carbon budget could be complex, with reduced productivity in the surface ocean potentially offset by changes in the efficiency of carbon export and remineralization at depth. The nitrogen cycle in oxygen minimum zones is also influenced by the activity of animals. Zooplankton and fish that migrate vertically through the oxygen minimum zone each day excrete ammonium, providing a source of nitrogen that can fuel amamics and nitrification. Some studies suggest that these migrating animals contribute significantly to the nitrogen budget of oxygen minimum zones, effectively transporting nitrogen from the surface waters where they feed to the low oxygen depths where they excrete. This is another example of the interconnectedness of ocean processes, the way that the movements and metabolisms of visible animals can influence the invisible workings of microbial communities. Scientists study the nitrogen cycle in oxygen minimum zones using a combination of field measurements, laboratory experiments, and computer models. They measure the concentrations of nitrogen compounds in the water, the rates of denitrification and amamics, the isotopic signatures that reveal the pathways through which nitrogen is transformed. They culture microbes in the lab, studying their metabolisms and their responses to changing conditions. They build models that simulate the physical and biological processes in oxygen minimum zones, testing hypotheses about how the nitrogen cycle might change in response to warming, deoxygenation, or other perturbations. What they have learned is that the nitrogen cycle in oxygen minimum zones is more complex and more dynamic than previously thought. It is not a simple linear process, but a network of interactions, feedbacks, and dependencies that respond to the environment in ways that are still being unraveled. Each new discovery reveals another layer of complexity, another way in which the ocean surprises us with its capacity for transformation and adaptation. Oxygen minimum zones are not distributed evenly across the ocean. They occur in specific regions where the combination of physical and biological factors creates the conditions for oxygen depletion. The largest and most intense oxygen minimum zones are found in the tropical and subtropical oceans. particularly along the eastern boundaries of ocean basins, where upwelling brings nutrient-rich water to the surface and drives high productivity. The eastern tropical Pacific hosts one of the most expansive oxygen minimum zones on Earth, stretching from the coast of Mexico to the waters off Peru. This zone encompasses millions of cubic kilometers of water with oxygen concentrations that in places drop to near zero. The waters here are warm, stratified, and rich in nutrients brought to the surface by upwelling. Phytoplankton blooms proliferate, and as they die, their remains sink into the oxygen minimum zone below, feeding the microbial communities that consume oxygen and drive nitrogen cycling. The intensity of this oxygen minimum zone is striking. In some regions, oxygen concentrations are so low that they fall below the detection limits of standard sensors, creating a truly anoxic core where only the most specialized microbes can survive. The Eastern Tropical Pacific Oxygen Minimum Zone is not a single uniform feature, but a complex of interconnected regions, each with its own characteristics. Off the coast of Peru, where the Humboldt Current brings cold, nutrient-rich water to the surface, the oxygen minimum zone comes close to the continental shelf, creating conditions where low oxygen water intersects with the seafloor. Here, microbial mats cover the sediment, and strange communities of sulfur-oxidizing bacteria thrive in the interface between the anoxic water and the sediment. Off Mexico, the oxygen minimum zone extends far offshore, creating a vast region of low oxygen water that shapes the distribution of marine life across the eastern Pacific. The Arabian Sea is another major oxygen minimum zone, shaped by the seasonal monsoon winds that drive intense upwelling and productivity. During the southwest monsoon, strong winds blow across the Arabian Sea, pushing surface water away from the coast and drawing deeper, nutrient-rich water upward. This fuels enormous phytoplankton blooms, and as in the Pacific, the sinking organic matter feeds the microbial communities in the oxygen minimum zone below, driving oxygen depletion. The Arabian Sea oxygen minimum zone is particularly intense, with large regions where oxygen is completely exhausted. It is also relatively shallow, in some areas coming to within 100 meters of the surface, making it one of the shallowest anoxic zones in the open ocean. The Arabian Sea oxygen minimum zone is also notable for its seasonal variability. During the southwest monsoon, when upwelling is most intense, the oxygen minimum zone expands and intensifies, driven by the increased productivity and the enhanced export of organic matter to depth. During the northeast monsoon, when upwelling weakens, the oxygen minimum zone contracts, and oxygen levels can recover somewhat as mixing and ventilation improve. This seasonal cycle creates a dynamic environment, where oxygen levels fluctuate over the course of the year and where marine organisms must adapt to these changing conditions. Off the coast of West Africa, another oxygen minimum zone exists, though smaller and less intense than those in the Pacific and Arabian Sea. This zone is associated with the Benguela upwelling system, where nutrient-rich water rises along the coast of Namibia and Angola, driving productivity and creating conditions for oxygen depletion in the subsurface waters. The Benguela oxygen minimum zone is subject to variability, expanding and contracting in response to changes in upwelling intensity and ocean circulation. In some years, strong upwelling drives intense productivity and severe oxygen depletion, creating dead zones along the coast where fish kills occur and fisheries are disrupted. In other years, upwelling is weaker and oxygen levels remain relatively high. The Benguela oxygen minimum zone also exhibits interesting spatial patterns. Near the coast, where upwelling is strongest and productivity is highest, oxygen depletion can be severe, creating anoxic conditions in the water column and on the seafloor. Further offshore, oxygen levels are higher, and the oxygen minimum zone is less intense. This creates a gradient of oxygen concentration from the coast to the open ocean, which influences the distribution of marine life and the structure of benthic communities. These major oxygen minimum zones are all located in tropical or subtropical regions, where warm temperatures and high productivity combine to create the conditions for severe oxygen depletion. But smaller, less intense oxygen minimum zones can be found in other parts of the ocean as well. In the North Pacific, a broad oxygen minimum zone exists at intermediate depths, though it is not as intensely depleted as the tropical zones. In the South Atlantic, oxygen concentrations at mid-depths are relatively high, and true oxygen minimum zones are rare. The distribution of oxygen minimum zones is not static. It changes over time, responding to shifts in ocean circulation, climate, and productivity. During glacial periods, when ocean circulation patterns were different and temperatures were cooler, oxygen minimum zones likely had different shapes and intensities than they do today. Sediment cores reveal evidence of past oxygen minimum zones, layers of organic-rich sediment that were deposited when bottom waters were oxygen depleted, preserving a record of ancient low-oxygen conditions. In the modern ocean, oxygen minimum zones appear to be changing in response to human activities. Climate change is warming the ocean, reducing the solubility of oxygen in seawater and increasing stratification, both of which contribute to deoxygenation. At the same time, nutrient pollution from agriculture and sewage is increasing productivity in coastal waters, leading to the formation of dead zones, regions where oxygen is depleted to the point that most marine life cannot survive. These dead zones are related to oxygen minimum zones but are driven by human inputs rather than natural ocean processes. The size and intensity of oxygen minimum zones in the open ocean have also changed over the past several decades. Measurements of oxygen concentration in the ocean, collected over the past 60 years, show a marked decline in oxygen content in many regions, particularly at mid-depths where oxygen minimum zones are located. This decline is associated with warming and with changes in ocean circulation that reduce the ventilation of deep waters. Open ocean areas with very low oxygen have expanded by more than 1.5 million square miles in the last 50 years, and coastal waters have seen a tenfold increase in low oxygen areas over the same period. The expansion of oxygen minimum zones has significant implications for marine ecosystems and for the global climate. As oxygen minimum zones grow, they compress the habitat available to fish and other animals that require higher oxygen levels. This can lead to changes in the distribution and abundance of commercially important species, affecting fisheries and the livelihoods of people who depend on them. It can also alter the structure of marine food webs, favoring species that are tolerant of low oxygen at the expense of those that are not. The expansion of oxygen minimum zones also affects the chemistry of the ocean, increasing nitrogen loss and nitrous oxide production, as discussed earlier. As more water becomes oxygen depleted, the biogeochemical cycles that regulate nutrients and greenhouse gases are altered, with potential consequences for ocean productivity and climate. Understanding these changes, predicting how they will unfold in the future, and assessing their impacts on ecosystems and human societies are major challenges for ocean science. To study oxygen minimum zones, scientists use a variety of tools and approaches. They deploy sensors on moorings and autonomous vehicles to measure oxygen concentration, temperature, salinity, and other properties continuously over time. They collect water samples from research vessels, analyzing them for nutrients, nitrogen compounds, isotopes, and microbial communities. They use genetic techniques to identify the bacteria and archaea present in oxygen minimum zones and to understand their metabolic capabilities. They conduct laboratory experiments to measure rates of denitrification, anemox, and other processes under controlled conditions. And they build computer models that simulate the physics, chemistry, and biology of oxygen minimum zones, testing hypotheses and making predictions about how these zones might change in the future. What emerges from all of this research is a picture of oxygen minimum zones as dynamic, complex, and globally significant features of the ocean. They are not static layers of low oxygen water, but regions shaped by ongoing processes, by the circulation of the sea, by the sinking of organic matter, by the activity of microbes, and by the changing climate. They are windows into the biochemical cycles that govern the ocean, into the limits of life, and into the ways that human activities are altering the fundamental workings of the Earth system. The ocean is changing. It is warming, acidifying, losing oxygen. These changes are driven by the accumulation of carbon dioxide in the atmosphere, a consequence of burning fossil fuels, deforestation, and other human activities. As the atmosphere warms, heat is transferred to the ocean, where it alters the physical and chemical properties of seawater. Warmer water holds less dissolved oxygen, and increased stratification reduces the mixing that replenishes oxygen in the deep. The result is deoxygenation, a slow suffocation of the ocean that is already underway and projected to intensify in the coming decades. Oxygen minimum zones are expanding. This is not speculation but observation. Measurements of oxygen concentration in the ocean, collected over the past 60 years, show a clear trend of declining oxygen levels at mid-depths, particularly in the regions where oxygen minimum zones exist. the extent of low oxygen water has increased, and the intensity of oxygen depletion in the cores of these zones has become more severe. This expansion is consistent with what we would expect from warming and increased stratification, and it is projected to continue as the climate warms further. Climate models predict that by the end of the 21st century, under scenarios of continued high greenhouse gas emissions, the volume of oxygen-depleted water in the ocean could increase by millions of cubic kilometers. In the tropical Pacific, one of the most intensively studied regions, models project that the oxygen minimum zone will expand both vertically and horizontally, growing by 6 to 8 million cubic kilometers, an increase of about 4%. This expansion will not be uniform. The outer layer of the oxygen minimum zone, where oxygen levels are low but not completely exhausted, will expand more than the core, where oxygen is already near zero. In fact, some models predict that the anoxic core of the oxygen minimum zone may actually shrink slightly, even as the overall zone expands. This diverging fate of the core and the outer layer is due to the complex interplay of factors that control oxygen minimum zones. Warming increases stratification, which reduces ventilation and causes the outer layer to expand. But it also reduces the supply of organic matter sinking from the surface, because warmer water can support less primary productivity in some regions. With less organic matter to fuel respiration, the intensity of oxygen depletion in the core may decrease. This is a small consolation, however, because the expansion of the outer layer means that a larger volume of water will be oxygen depleted, and the habitat available to animals that require higher oxygen levels will shrink. The expansion of oxygen minimum zones will have cascading effects on marine ecosystems. Many commercially important fish species, such as tuna, swordfish, and marlin, are sensitive to low oxygen and avoid oxygen minimum zones. As these zones expand, the vertical range of habitat available to these fish will compress, squeezing them into a narrower layer near the surface. This habitat compression can make fish more vulnerable to overfishing because they are concentrated in a smaller volume of water and thus easier to catch. It can also affect their physiology and behavior, forcing them to spend more time in suboptimal conditions or to migrate to areas where oxygen levels are more favorable. Other species may benefit from the expansion of oxygen minimum zones, at least in the short term. Species that are tolerant of low oxygen, such as jellyfish, certain squid, and some fish, may expand their ranges as oxygen minimum zones grow. The shifting balance of species, with low oxygen tolerant species increasing and high oxygen requiring species declining, could fundamentally alter the structure of marine food webs, with unpredictable consequences for ecosystem function and the services that oceans provide to humans. The expansion of oxygen minimum zones will also affect the chemistry of the ocean and the climate. As more water becomes oxygen depleted, the loss of nitrogen through denitrification and anammox is likely to increase, further limiting ocean productivity. At the same time, the production of nitrous oxide, a potent greenhouse gas, may increase, contributing to additional warming and creating a positive feedback loop. The magnitude of this feedback is uncertain, and it depends on the details of how oxygen minimum zones change and how microbial communities respond. But it is a potential tipping point, a way in which changes in the ocean could amplify climate change. Not all regions of the ocean will experience the same changes. In some areas, changes in circulation may actually improve ventilation and reduce oxygen minimum zone intensity, at least temporarily. The Southern Ocean, for instance, plays a critical role in supplying oxygen to the deep ocean, and changes in wind patterns or mixing in this region could alter the supply of oxygen to oxygen minimum zones elsewhere. There is also evidence that ocean circulation patterns can shift abruptly in response to warming, potentially causing rapid changes in oxygen minimum zone extent and intensity. These abrupt changes are difficult to predict, and they add uncertainty to projections of how oxygen minimum zones will evolve in the future. Scientists are working to improve their understanding of oxygen minimum zones and their response to climate change. They are collecting more data, deploying new sensors, refining computer models, and conducting experiments to test hypotheses about how oxygen minimum zones form and change. They are also looking to the past, studying sediment cores and other geological records to understand how oxygen minimum zones have responded to past climate changes. The geological record reveals that oxygen minimum zones have expanded and contracted many times over Earth's history, sometimes growing to much larger sizes than today, and sometimes shrinking to near absence. These past changes were driven by shifts in climate, ocean circulation, and productivity, and they provide clues about how oxygen minimum zones might respond to future warming. One of the lessons from the geological record is that oxygen minimum zones can change rapidly, on timescales of decades to centuries when the right conditions are met. This raises the possibility that the expansion of oxygen minimum zones we are seeing today could accelerate, particularly if warming triggers changes in ocean circulation or other tipping points in the Earth system. The risk of rapid change underscores the urgency of reducing greenhouse gas emissions and mitigating climate change, to avoid pushing the ocean into a state that is fundamentally different from the one we know. Research conducted over the past decade has begun to reveal the mechanisms by which warming drives deoxygenation. When the surface ocean warms, the difference in density between surface and deep water increases, making the thermocline more stable and reducing vertical mixing. This means that less oxygen-rich surface water is transported downward, and less oxygen-depleted deep water is brought upward to be refreshed at the surface. The ocean becomes more stratified, more layered, and the exchange of oxygen between the atmosphere and the deep ocean slows. At the same time, warmer water holds less dissolved oxygen. The solubility of gases in water decreases with increasing temperature, a basic principle of chemistry that has profound implications for the ocean. As the upper ocean warms, even if mixing remained the same, oxygen concentrations would decline simply because warm water cannot hold as much oxygen as cold water. The combination of reduced solubility and reduced mixing creates a double impact on oxygen levels, driving deoxygenation across large regions of the ocean. These changes are not uniform across the ocean. Some regions are more vulnerable to deoxygenation than others, depending on local circulation patterns, stratification, and productivity. The tropical oceans, where oxygen minimum zones are already most pronounced, are particularly susceptible. Here, the water is already warm and stratified, and even small increases in temperature can have large effects on oxygen levels. The North Pacific, with its sluggish circulation and large oxygen minimum zone, is also at risk. The Atlantic, with its more vigorous circulation, may be more resilient, but it too is experiencing deoxygenation in some regions. Scientists are also concerned about potential tipping points, thresholds, beyond which small changes in forcing could lead to large, rapid changes in oxygen minimum zones. One such tipping point could occur if warming disrupts the circulation patterns that currently ventilate the deep ocean. The formation of deep water in polar regions, which currently supplies oxygen to the deep ocean, could be affected by warming and freshening of surface waters. If deep water formation slows or stops, the supply of oxygen to the deep ocean could be cut off, leading to rapid expansion of oxygen minimum zones and potentially to widespread anoxia. Another potential tipping point involves the positive feedback between deoxygenation and productivity. As oxygen minimum zones expand, nitrogen loss increases, which reduces the availability of nitrogen for phytoplankton growth. This could lead to reduced productivity in the surface ocean, which would reduce the export of organic matter to depth, which could in turn reduce oxygen consumption and slow the expansion of oxygen minimum zones. But the relationship is complex, and it is also possible that changes in the nitrogen cycle could amplify deoxygenation rather than mitigate it, depending on the specifics of how productivity and nitrogen cycling respond to warming. The future of oxygen minimum zones is not yet written. It depends on the choices we make today, on whether we continue to emit greenhouse gases at current rates or take action to reduce emissions and stabilize the climate. It also depends on natural variability in the ocean, on the complex feedbacks and interactions that govern oxygen minimum zones, and on factors that we may not yet fully understand. But the trajectory is clear. Without action, oxygen minimum zones will continue to expand. The ocean will continue to lose oxygen, and the consequences for marine life and the climate will be profound. The expansion of oxygen minimum zones is not an abstract problem for the future. It is already affecting marine ecosystems and the people who depend on them. In the eastern tropical Pacific, off the coasts of Peru and Chile, oxygen minimum zones have been expanding and intensifying for decades, compressing the habitat available to fish and altering the distribution of commercially important species. In the Arabian Sea, the oxygen minimum zone has shoaled, coming closer to the surface and encroaching on the depths where fish normally feed and reproduce. These changes are measurable, documentable, and consequential. For fish and other mobile animals, the most immediate impact of oxygen minimum zone expansion is habitat compression. Fish that require high oxygen levels to support their active lifestyles are squeezed into a narrower vertical band, trapped between the warm, oxygen-poor surface waters and the oxygen-depleted depths below. This compression reduces the three-dimensional space available to them, potentially increasing competition for food and space and making them more vulnerable to predation and fishing. This habitat compression has been documented in multiple regions and for multiple species. Studies using electronic tags to track fish movements have revealed that as oxygen minimum zones shoal and expand, fish spend more time in shallow waters, closer to the surface, where they are more exposed to fishing gear and where environmental conditions may be suboptimal. During the day, when surface waters warm under the tropical sun, fish may be caught between the need to avoid the heat and the need to avoid the oxygen minimum zone below, creating a squeeze that limits their options and forces them into a narrow band of tolerable conditions. Blue marlin, for instance, are large, fast-swimming predators that hunt in the upper ocean but need access to deeper waters for feeding and thermoregulation. Studies using electronic tags to track the movements of blue marlin in the Atlantic have shown that as oxygen minimum zones shoal, the depth range available to these fish shrinks. Between 1960 and 2010, the oxygenated upper layer of the tropical northeast Atlantic thinned at a rate of about 1 meter per year, leading to a 15% loss of habitat for species like blue marlin. This habitat loss makes these fish more vulnerable to surface fishing gear, as they are forced to spend more time in shallow waters where they are more likely to encounter fishing lines and nets. The compression of habitat does not just affect individual fish, it affects entire populations, and can alter the structure of marine food webs. When predators like tuna and marlin are squeezed into a narrower depth range, they may encounter their prey more frequently, which could increase predation pressure on smaller fish and squid. Alternatively, if the predators are themselves stressed by the low oxygen or high temperatures, their feeding efficiency may decline, reducing predation pressure. The outcome depends on the specific circumstances, on how different species respond to the changing conditions, and on the complex interactions between predators, prey, and the physical environment. Tuna face similar challenges. Yellowfin and big-eye tuna are highly migratory species that travel vast distances across the Pacific and Atlantic, hunting for prey in the surface waters, but also diving deep to feed on squid and other organisms. Oxygen minimum zones present a barrier to these deep dives, limiting how far down the tuna can go without encountering oxygen levels that are too low to sustain their metabolic demands. As oxygen minimum zones expand, the depth range accessible to tuna shrinks, potentially affecting their feeding success and their distribution across the ocean. Research has shown that big-eye tuna, in particular, are sensitive to oxygen levels and spend much of their time near the lower boundary of the oxygen minimum zone, where temperatures are cooler, but oxygen is still sufficient for their needs. As the oxygen minimum zone expands upward, this boundary moves closer to the surface, compressing the thermal habitat of the tuna and potentially affecting their ability to thermoregulate and to avoid warm surface waters during the day. This can have consequences for their physiology, their growth rates, and their reproductive success. The effects of oxygen minimum zone expansion are not limited to large predatory fish. Smaller fish, squid, and crustaceans are also affected, with some species declining and others expanding as oxygen levels change. Anchovies and sardines, which are among the most abundant fish in the eastern Pacific, show distribution patterns that are strongly influenced by oxygen concentration. These small pelagic fish tend to avoid the core of the oxygen minimum zone, concentrating in the well-oxygenated surface waters or moving to regions where oxygen levels are higher. As oxygen minimum zones expand, the distribution of these fish changes, becoming more concentrated near the coast or in areas where upwelling brings oxygen-rich water to the surface. Zooplankton, the tiny drifting animals that form the base of the marine food web, show vertical distribution patterns that are strongly influenced by oxygen concentration. As oxygen minimum zones expand, zooplankton communities shift, with some species avoiding low oxygen waters and others taking advantage of the reduced predation pressure in these zones. These shifts in zooplankton distribution can cascade through the food web, affecting the availability of food for fish and altering the structure of marine ecosystems. Fisheries are already feeling the effects of oxygen minimum zone expansion. In the eastern Pacific, fisheries for anchovies, sardines, and other small pelagic fish are some of the most productive in the world, supporting livelihoods for millions of people. These fisheries depend on the upwelling that brings nutrients to the surface, fueling the phytoplankton blooms that feed the fish. But upwelling also drives the formation of oxygen minimum zones, and as these zones expand, the distribution of fish changes, becoming more concentrated near the coast or in areas where oxygen levels are higher. This can make the fish easier to catch in the short term, but it also makes the fisheries more vulnerable to collapse if fishing pressure is not carefully managed. In the Arabian Sea, fisheries for tuna, mackerel, and other species are similarly affected by changes in oxygen levels. The shoaling of the oxygen minimum zone has compressed the habitat available to these fish, altering their distribution and potentially affecting their abundance. For the people who depend on these fisheries for food and income, the changes are not abstract. They are visible in the size and composition of the catch, in the effort required to find fish, and in the economic uncertainty that comes with a changing ocean. The expansion of oxygen minimum zones also affects the benthic communities, the organisms that live on or in the seafloor. Where oxygen minimum zones intersect the seafloor, typically on the continental shelf or upper slope, the low oxygen levels can create conditions that are hostile to most benthic life. In these areas, the seafloor may be dominated by microbial mats, thick layers of bacteria that can tolerate or even thrive in anoxic conditions. These mats are fundamentally different from the diverse benthic communities found in well-oxygenated regions, which include worms, mollusks, crustaceans, and other animals that burrow into the sediment and play important roles in nutrient cycling and sediment mixing. The transition from a diverse benthic community to a microbial mat is not gradual. It can happen relatively quickly when oxygen levels drop below a critical threshold, leading to mass mortality of benthic animals and a shift to a microbial-dominated ecosystem. This shift has been observed in several regions where oxygen minimum zones have expanded or intensified, and it represents a fundamental change in the ecology and biogeochemistry of the seafloor. The ecological impacts of oxygen minimum zone expansion are not limited to direct effects on individual species or communities. They also include indirect effects, mediated through changes in food web structure, predator-prey interactions, and ecosystem function. For example, if oxygen minimum zones compress the habitat of large predatory fish, this may reduce predation pressure on smaller fish, allowing their populations to increase. But this increase may in turn affect zooplankton populations, which may decline if grazing pressure increases. These cascading effects can ripple through the entire food web, leading to changes in ecosystem structure that are difficult to predict and may have unforeseen consequences. Understanding the impacts of oxygen minimum zone expansion on marine ecosystems requires not only studying individual species and processes, but also taking a holistic ecosystem-level approach. Scientists are using a variety of tools to do this, including field observations, laboratory experiments, and computer models that simulate the interactions between physical, chemical, and biological processes in the ocean. They are also looking to the past, studying how marine ecosystems responded to changes in oxygen levels during previous periods of Earth's history, such as during oceanic anoxic events in the Mesozoic era. What emerges from this research is a picture of marine ecosystems that are resilient in some ways but vulnerable in others. Life in the ocean has adapted to a wide range of conditions over millions of years, and it has survived and thrived through periods of dramatic environmental change. But the rate of change we are seeing today, driven by human activities, is unprecedented in recent geological history. The question is not whether marine ecosystems can adapt to changing oxygen levels, but whether they can adapt quickly enough, and what the consequences will be for the species and the people who depend on them. Oxygen minimum zones are not a new phenomenon. They have existed in the ocean for as long as there have been oceans, waxing and waning in response to changes in climate, circulation, and productivity. The geological record, preserved in sediment cores and rock formations, reveals that oxygen minimum zones have varied dramatically over Earth's history, sometimes expanding to much larger sizes than today, and sometimes shrinking to near absence. These past changes provide a window into how oxygen minimum zones form, how they respond to environmental forcing, and what their consequences might be for marine life and the global climate. One of the most dramatic periods of oxygen depletion in Earth's history occurred during the Mesozoic era, particularly in the Cretaceous period, between about 145 million and 66 million years ago. During this time, The ocean experienced several oceanic anoxic events, periods when large portions of the ocean became depleted of oxygen, creating vast oxygen minimum zones that extended across entire ocean basins. These events were driven by a combination of factors, including intense volcanic activity, which released large amounts of carbon dioxide into the atmosphere, warming the climate and increasing stratification, and high rates of productivity, which led to increased oxygen consumption at depth. During oceanic anoxic events, the ocean's chemistry changed dramatically, with oxygen depleted, anaerobic processes dominated, and the ocean became enriched in hydrogen sulfide and other reduced compounds. The seafloor in oxygen-depleted regions became covered with organic-rich black shales, layers of sediment that were deposited under anoxic conditions and preserved a record of the event. These black shales are found in rock formations around the world, and they are one of the main sources of oil and gas formed from the organic matter that accumulated during these periods of oxygen depletion. The expansion of oxygen minimum zones during oceanic anoxic events had profound effects on marine life. Many species went extinct, unable to adapt to the low oxygen conditions, but others survived, and some even thrived, taking advantage of the opportunities created by the changing environment. For example, the ancestors of the modern vampire squid may have evolved their adaptations to low oxygen during one of these periods, allowing them to survive in oxygen minimum zones that expanded during the Mesozoic and have persisted, in various forms, ever since. The fossil record provides evidence for this evolutionary history. A recent discovery of a vampire squid fossil from the Oligocene epoch, about 30 million years ago, bridges a gap in the fossil record and suggests that vampire squids have been living in oxygen minimum zones for at least tens of millions of years. The fossil, found in what was once a deep ocean basin with low oxygen levels, shows that by the Oligocene, vampire squids had already adapted to bathyal, oxygen-depleted habitats. This suggests that the deep-sea specialization of vampire squids was triggered by the development of oxygen minimum zones during the Cretaceous and Cenozoic, as the ocean's chemistry and station changed in response to tectonic activity, climate shifts, and the evolution of life itself. The geological record also reveals that oxygen minimum zones have fluctuated over shorter timescales, on the order of thousands to tens of thousands of years, in response to changes in ocean circulation and productivity. During the last glacial period, for example, oxygen minimum zones in the Pacific and Atlantic were different in size and intensity than they are today, shaped by the different patterns of ocean circulation that prevailed under glacial conditions. Sediment cores from these regions contain layers of sediment that were deposited under varying oxygen conditions, providing a detailed record of how oxygen minimum zones have changed over time. By studying these past changes, scientists can gain insights into the factors that control oxygen minimum zones and how they might respond to future climate change. For example, the geological record shows that oxygen minimum zones tend to expand during periods of warming and high productivity, which is consistent with what we are seeing today. But it also shows that the response is not always linear or predictable. There are thresholds and tipping points where small changes in forcing can lead to large changes in oxygen minimum zone extent and intensity. Understanding these thresholds is critical for predicting how oxygen minimum zones might change in the future and what their impacts might be. The ancient ocean also provides examples of how life has adapted to oxygen minimum zones over evolutionary time. The microbes that dominate oxygen minimum zones today, performing denitrification, anaerobics, and other anaerobic processes, are descended from lineages that evolved billions of years ago, when the Earth's atmosphere and ocean were very different. Before the rise of oxygen in the atmosphere, around 2.5 billion years ago, the ocean was largely anoxic, and anaerobic metabolisms were the norm. The microbes that perform these metabolisms today are thus evolutionary relics, survivors from a time when the ocean was a very different place. As oxygen levels rose in the atmosphere and ocean, aerobic metabolisms became dominant, and anaerobic processes were relegated to environments where oxygen was scarce, such as sediments, oxygen minimum zones, and other low oxygen habitats. But these anaerobic microbes did not disappear. They persisted, adapting to the changing environment, and they continue to play critical roles in the biogeochemical cycles that govern the ocean and the planet. The nitrogen cycle, in particular, depends on these anaerobic processes, and without them, the ocean's chemistry would be fundamentally different. The study of ancient oxygen minimum zones is not just an academic exercise. It has practical implications for understanding the modern ocean and predicting its future. By looking to the past, scientists can test their models, refine their understanding of the processes that control oxygen minimum zones, and gain confidence in their predictions of how these zones might change in response to warming. The past is not a perfect analog for the future, because the rate of change today is much faster than in most periods of Earth's history, and because human activities are introducing novel forcings that have no direct parallel in the geological record. But the past provides context, a sense of the range of variability that the ocean can experience, and a reminder that the ocean has been through dramatic changes before and will likely continue to change in ways that we cannot fully anticipate. Studying oxygen minimum zones is a technical challenge. Oxygen is invisible, odorless, tasteless, and its concentration in seawater can vary by orders of magnitude over short distances. Traditional methods of measuring oxygen, such as the Winkler titration, are accurate but labor-intensive and can be contaminated by exposure to air during sample collection. Modern sensors, such as optical oxygen sensors and electrochemical sensors, have improved the ease and accuracy of oxygen measurements, but they still have limitations, particularly at very low oxygen concentrations where even small amounts of contamination can skew the results. One of the major challenges in studying oxygen minimum zones is sampling them without introducing oxygen contamination. When a water sample is collected from an oxygen minimum zone and brought to the surface, it passes through layers of oxygenated water, and there is a risk that oxygen from these layers, or from the atmosphere, will contaminate the sample. This is particularly problematic for studying anaerobic processes, which are extremely sensitive to oxygen. Even trace amounts of oxygen at concentrations of a few nanomolar can inhibit denitrification, anammox, and other anaerobic metabolisms, making it difficult to study these processes in their natural state. To overcome this challenge, scientists have developed new sampling techniques and instruments that minimize oxygen contamination. These include specialized water samplers that seal tightly to prevent contact with air, autonomous underwater vehicles that can collect samples at depth without bringing them to the surface, and in situ sensors that measure oxygen and other properties directly in the water without disturbing the environment. One particularly innovative approach is the use of in situ incubation chambers, which allow scientists to measure rates of denitrification, anammox, and other processes in the oxygen minimum zone itself without the need to bring samples to the surface. Another challenge in studying oxygen minimum zones is the variability in oxygen concentration over space and time. Oxygen levels can change rapidly in response to physical processes such as mixing, advection, and upwelling, as well as biological processes such as respiration and photosynthesis. This variability makes it difficult to get a complete picture of oxygen minimum zones based on discrete samples collected at specific times and locations. To address this, scientists are increasingly using autonomous instruments such as gliders and profiling floats that can measure oxygen and other properties continuously over time and space, providing a much more detailed view of oxygen minimum zones than was possible with traditional ship-based sampling. Remotely operated vehicles, or ROVs, have also revolutionized the study of oxygen minimum zones. These underwater robots, controlled from a ship at the surface, can dive to depths of several thousand meters and explore oxygen minimum zones directly, using cameras, sensors, and sampling devices to study the environment and the organisms that live there. ROVs have been instrumental in documenting the presence and behavior of animals in oxygen minimum zones, such as the vampire squid, and in collecting samples of microbial mats and other features that are difficult to study with traditional sampling methods. The study of oxygen minimum zones also relies heavily on computer models, which simulate the physical, chemical, and biological processes that control oxygen concentration in the ocean. These models range from simple box models, which represent the ocean as a series of interconnected reservoirs, to complex three-dimensional models, which simulate ocean circulation, temperature, salinity, and biogeochemical cycles at high resolution. By comparing the results of these models with observations from the field, scientists can test their understanding of oxygen minimum zones and make predictions about how they might change in the future. One of the key insights from modeling studies is that oxygen minimum zones are sensitive to a wide range of factors, including ocean circulation, stratification, productivity, and the efficiency of organic matter remineralization. Small changes in any of these factors can have large effects on oxygen minimum zone extent and intensity, making predictions uncertain. This uncertainty is one of the reasons why projections of future oxygen minimum zones vary widely across different models, and why continued observations and model improvements are essential for understanding and predicting ocean deoxygenation. Scientists are also using genetic and molecular techniques to study the microbes that inhabit oxygen minimum zones. By extracting DNA from water samples and sequencing it, they can identify the bacteria and archaea present in the oxygen minimum zone and infer their metabolic capabilities. This approach, called metagenomics, has revealed a remarkable diversity of microbes in oxygen minimum zones, including many species that were previously unknown. It has also provided insights into the genes and enzymes that these microbes use to perform denitrification, anammox, sulfur oxidation, and other processes, helping to connect microbial ecology with biogeochemistry. Another technique, called metaproteomics, goes a step further by identifying the proteins that microbes are actually producing, which provides a more direct measure of their metabolic activity. By combining genomics, proteomics, and traditional biogeochemical measurements, scientists are building a more complete picture of how microbes in oxygen minimum zones function and how their activity responds to changes in the environment. The study of oxygen minimum zones is an ongoing effort, with new discoveries being made regularly as new techniques and instruments become available. Each new dataset, each new model simulation, each new genomic sequence adds to our understanding of these complex and dynamic features of the ocean. And as we learn more, we gain the ability to predict how oxygen minimum zones might change in the future and what the consequences might be for marine life, for biogeochemical cycles, and for the climate. We cannot see oxygen minimum zones from the surface. We cannot feel them beneath the keel of a ship or detect them in the taste of seawater. They are hidden, buried in the middle layers of the ocean, detectable only through careful measurement and inference. And yet they are vast, occupying millions of cubic kilometers of the ocean, shaping the distribution of life, influencing the chemistry of the sea, and playing a role in the regulation of the Earth's climate. In these zones, the ocean holds its breath, or rather, it holds what little breath it has left. Oxygen concentrations drop to levels so low that most life cannot survive, creating a world that is alien even by the standards of the deep sea. And yet, life persists. It persists in the form of microbes, invisible but numerous, performing biochemical transformations that have global consequences. It persists in the form of specialized animals, like the vampire squid, that have learned to thrive in conditions that would be lethal to their relatives. It persists in the form of migrants, fish and zooplankton that pass through these zones each day, enduring the hypoxia as a necessary part of their daily routines. The oxygen minimum zone is a place of limits, a testing ground for the boundaries of life, but it is also a place of opportunity, a refuge for those that can adapt, a source of nutrients and energy for those that have the right biochemical tools. It is a reminder that the ocean is not a single homogeneous environment, but a mosaic of habitats, each with its own conditions, its own challenges, its own communities of life. The expansion of oxygen minimum zones in response to climate change is a warning. It is a sign that the ocean is changing in fundamental ways, that the balance of oxygen and carbon and nitrogen that has prevailed for thousands of years is shifting. The consequences of this shift are not yet fully known, but they are likely to be profound, affecting marine ecosystems, fisheries, and the global climate. Understanding oxygen minimum zones, predicting their future, and mitigating their expansion are among the most important challenges facing ocean science today. As we float here, suspended in thought, the ocean continues its slow work. Water circulates, particles sink, microbes respire, and oxygen fades into scarcity. The oxygen minimum zone persists, a layer of near suffocation maintained by forces that operate on scales from the microscopic to the global, from seconds to millennia. It is a hidden feature of the ocean, but its influence is felt far beyond its boundaries, in the chemistry of the sea, in the distribution of life, and in the changing climate of the planet. And so, as the night deepens and sleep draws near, let yourself drift with the currents of the sea, down through the layers of oxygen and scarcity, into the twilight zone where the vampire squid glides through the darkness, where microbes perform their invisible work, where the ocean slowly, patiently, holds what little breath remains. And so, as the night deepens and sleep draws near, Let yourself drift with the currents of the sea, down through the layers of oxygen and scarcity, into the twilight zone where the vampire squid glides through the darkness, where microbes perform their invisible work, where the ocean slowly, patiently, holds what little breath remains. Think of the ocean not as a single entity, but as a collection of interconnected systems, each influencing the others in ways that ripple across scales of space and time. The oxygen minimum zones we have explored tonight are not isolated features, but part of the global ocean circulation, connected to the polar seas where deep water forms, to the tropical upwelling zones where nutrients rise to the surface, to the vast gyres that spin slowly across ocean basins. What happens in one part of the ocean affects all the others, through currents and winds and the exchange of heat and gases at the surface. The oxygen you breathe on land, produced by plants and phytoplankton, is connected to the oxygen that vanishes in the oxygen minimum zone. The nitrogen that limits plant growth on land and in the sea is cycled through the oxygen minimum zone, where it is transformed and released back to the atmosphere. The carbon dioxide that accumulates in the atmosphere, warming the planet, affects the ocean's temperature and chemistry, which in turn affects oxygen levels and the extent of oxygen minimum zones. Everything is connected, everything is in motion, and the ocean, vast as it is, responds to even small changes in the balance of these forces. As you rest tonight, consider the invisible architecture of the sea, the layers and gradients and boundaries that structure the ocean's interior. Consider the creatures that have adapted to life at the limits of possibility, breathing the thinnest air, surviving in places where most life cannot exist. Consider the microbes that have been performing their chemical transformations for billions of years, long before humans walked the earth, and will likely continue long after we are gone. Consider the ocean's patience, its slow rhythms, its capacity to absorb and respond to change over timescales that dwarf human lifetimes. The oxygen minimum zones are expanding, yes, but they are also teaching us. They are teaching us about the limits of life, about the biochemical creativity of evolution, about the interconnectedness of ocean processes, and about the consequences of altering the composition of the atmosphere. They are teaching us that the ocean is not an inexhaustible resource, not an unchanging backdrop to human activity, but a dynamic system that responds to forcing, that has thresholds and tipping points, and that can change in ways that affect all life on Earth. In the deep twilight of the oxygen minimum zone, where light has faded and oxygen has vanished, life continues in forms both familiar and strange. The vampire squid drifts, patient and slow, its enormous eyes watching for the next piece of marine snow. The bacteria transform nitrogen, cycling it back to the atmosphere in a process that has shaped the chemistry of the ocean for eons. The water flows, carrying with it the signatures of distant sources and the promise of eventual renewal. And the ocean, vast and deep and full of mysteries, continues its ancient work, indifferent to our presence, shaped by forces we are only beginning to understand. Rest now, carried by the deep rhythms of the sea, knowing that even in the places where oxygen fades, life finds a way and the ocean endures, vast and patient and full of mysteries still waiting to be explored. Let the gentle currents of sleep carry you deeper, past the last whispers of breath, into the quiet darkness where the sea holds its secrets. Good night.